giving life, or we're giving technology lifelike qualities to such an extent that as humans, we almost kind of recognize the fact, and actually many of us did recognize the fact, that the technology becoming so complex that we have no way to fully control it. So it's already beyond our ways of controlling it. It does have a life of its own. At the very same time that we are trying to make life more and more like a technology. I talk about it in terms of psychopathologies of control, that we relinquish control over systems that have been developed by humans from the bottom up, and we're now obsessed with trying to control systems that existed for millions of years and billions of years outside of our control, and extremely complex systems, and we're trying to figure out ways to make them into our servants, to control them. And again, this is something that we need to keep in mind when we think about kind of our ability and our attempts and the mindset that biodesign, for example, represents. Another really interesting thing that happened to me when I was starting to do the research, or continuing the research. So in 1995, in the midst of my studies, this image hit the media. So this is, hopefully all of you seen it before. This is, from my perspective, as someone who self-represented or self-identify as an artist, this image is one of the most important images of the late 20th century. It's really, the surrealist dream comes alive, but it comes alive through the hands of scientists and not artists. That was developed by a group of tissue engineers in Boston, in a many, basically as a means of highlighting the powers of those new technologies, and as a way to attract attention, even though there's some stories. I interviewed all of the scientists who were involved. Most of the people who were involved in the project were not interested at the time in 1995 to present it publicly, but one of them, Professor Charles Vacanti, decided to show it to a BBC documentary filmmaker, and that became kind of the poster kid of tissue engineering. But also it represented the ability to start to sculpt with living biological materials to such an extent as to bring into life some of kind of the deep-rooted fantasies that we humans seem to share, and that's this idea of the chimera, the idea of the human-animal hybrid that seems to be represented in most human mythologies. And here it is, kind of alive and kicking on what becomes kind of the new stage of the drama that is unfolding in regard to life, and that's the Petri dish. So we have this kind of what wasn't intended to be an artwork, having a much more significant power and a much stronger hold on our imagination than any artwork can do. And here we see the beginning of this collapse of those social contracts that I referred to before. So this is kind of the publicity image, or the image from the BBC documentary. Those are the images of the actual scientific paper that was eventually published in 1997. So you can see the staging is very different. The lighting is different, not as impressive, but they're still very striking images of those surrealist mice that are around there. So what I decided to do, I decided to conduct my research and look at the potential future where design and biotechnology might interact. And I just recently refound this letter that I decided to send to people when I was doing my research, to try and help me understand what I'm dealing with. And as you can see already, I did a really horrible logo. Oh, trying to move that's stuck. Okay, there is the logo here. If you can see that I done actually referring to Biodesign. So this is from 1996, the letter that was sent with Oron Biodesign, trying to push this field with, as I mentioned before, a somewhat naive perspective. But you might be able to see it, might be too small for you. But the first question that I asked those experts that I approached was, what are the ethics, what are the ethical issues that and the public acceptance of bioengineered products in any level of usefulness? I talked about this idea of generating public discussion in regard to other uses of biotechnology, and to try to conceptualize ways in which I can put my skills as a designer to use in developing new bioengineered products, and then looking at the design and its boundaries and its limits, already kind of recognizing the collapse of those contracts, and the use of biotechnology products in architecture. So this is 1996, very, very naive, without much understanding of where things are. And obviously, as far as I know, and my research indicated, there was very little, if at all, research that was done into the use of living biological materials and biotechnologies for the purpose of designing consumer products or products which are other than agricultural and medical. So again, just kind of, obviously there were some statements and there were some kind of ideas about the notion that life can become a raw material. This is a quote from A.G. Wells from 1895, just before he wrote The Island of Dr. Moreau. And he said there, what is obviously not the case anymore, that we overlook only too often the fact that a living being may also be regarded as a raw material, as something plastic, that's something that may be shaped and altered. And I would argue that now every living being is seen as a potential resource and an asset to be exploited, and we're not overlooking anymore the fact that those things can be regarded as raw materials. For better or for worse, it's those are exactly the question that we need to start to address. So as my research progressed, I decided to look at one technology in particular as kind of my outlet to start to raise some of the issues that I felt need to be addressed. I should tell you that I graduated from my product design degree and never really practiced as a product designer, because I decided that it's much more important to continue my research as an artist, where I can open up problems and questions rather than engage in a solutionist type of approach and try to figure out how we can make products out of living things. And my kind of technology of choice was tissue culture and tissue engineering, because I felt that raises quite a lot of really interesting issues in regard to deep-held notions of what a body is. If you start to fragment bodies, if you start to grow fragments and parts of complex bodies outside of the original body from which they were taken, it raises much more interesting questions and kind of highlight what I refer to as our poverty of language, in the sense that we are lacking any cultural tools, or almost completely lacking cultural tools to deal with what is coming in regard to our new mindset or new approaches to life. So the first person who was credited as kind of really institutionalizing the idea of keeping fragments of complex bodies alive outside of the original body from which they were taken is Alexis Carrel. And you can see the type of imagery that already been presented as he was starting to publicize his work. So we're talking about the early 20th century, and here you see kind of a caricature where you have those different types of chimeras all produced and conjured by this new magician, Alexis Carrel, the scientist and the alchemist, in a sense, that was starting to maintain and grow those fragments of bodies. So already kind of the public imaginary started to think about those in those chimerical kind of terms. One of his colleagues in 1916 already recognized the fact that through the discovery of tissue culture, they didn't just find a way to grow cells outside of the original body, they did create a new type of body, which is the artificial technoscientific body in which to grow the cell. And this new body, this new technoscientific body, is a really important part of the conversation, because this is what often is being removed from the discussions when we are now thinking about kind of growing living biological products as a way of solving problems in the world. Those are things that are heavily artificial and require lots of resource, but also conceptually exist in a place where we don't really think a lot about. And one of the greatest examples of that is the fact that when baby incubators have been kind kind of brought to the United States back around the same time that Carrel was doing his initial tissue culture experiments. Those baby incubators were presented within the freak show initially at the Pan American Expo in Buffalo, but later at Coney Island. So you can see infant incubators with living infants. That operation moved to Coney Island, and now you can see the Coney Island installation, and you can see also that the distributors of those baby incubators was still coming from Dreamland, Coney Island, and distributed for both hospitals and amusement parks. Now, there's many reasons for that, but in the context of what I'm trying to say, the fact that you had little human bodies embedded in a techno-scientific body in the incubator, this combination between bodies and technologies had no place to be put within the cultural context, not even a hospital initially, and the only cultural space that they could occupy was the freak show. And that's because of their inability to articulate the existence of those human-machine hybrids, in a sense, at the time. So, and to a large extent also, those living infants had to earn their living by actually being put on display as a way of kind of allowing them to survive. And those incubators had about 80% success rate, as opposed to 0 if those babies had been left alone. But it took a really long while for the medical establishment and culture in general to take them away from the context of the freak to something which is much more established. And to a large extent, some of what's happening at the moment can be seen as a very similar approach, that those things are still in the realm of those oddities, those freaks, and they're not really yet or at all being kind of put within the mainstream idea of what we can do to living systems. And in a sense, the shift from artists working with biotechnology to designers working with biotechnology is part of this normalization process and the removal of those objects from the realm of the freak to the realm of the usefulness. So what's quite interesting for me, starting as a designer, was why no designers picked up on those biotechnologies until the mid 2000s. Yeah, so there was a flurry of artistic activity taking place in the mid 90s around those fields, but very, very little attention from the design world. And that shifted quite rapidly about a decade later when designers started to pick up. And we need to understand that it's kind of interesting to see also, because many of those technologies we're talking about existed within the biomedical and biotechnological realm for many, many years. But the 90s was a really interesting time. Obviously, there was the Human Genome Project that started in the late 80s as kind of the biggest scientific endeavor to map the human genome. And the type of promises and the hype that surrounded the Human Genome Project was particularly important in the sense that it gave a sense that once we have the human genome deciphered, we will be able to basically build a human from scratch. The idea was then that DNA is basically like a blueprint, and once we have it, we have the full instruction book for the construction of bodies, obviously in this case human bodies. Now they announced the completion of the so-called first draft of the Human Genome Project back in the year 2000, and it was celebrated all over the world. And with statements like this project was so someone from the director of the Wellcome Trust said a few months ago, I compared the project to the invention of the wheel. On reflection, it's way more than that. This code is the essence of mankind. Yeah, so this is the type of rhetoric that was used. But exactly a year later, they considered the fact that that original announcement in the year 2000 was not just premature, but also that deducted more by political than scientific criteria, and that the announcement was more grandiose than the situation warranted. But the hype, the narrative, the promises, and obviously the public imagination in regard to our power over biological systems very much stayed there as something that we fantasize about. It's also not a coincidence that at the very same time that the Human Genome Project was going on, also the very first FDA-approved GMO food was put on the market, and that's the Flavor Saver tomato, which had a gene from a fish inserted into a tomato to allow it to have a longer shelf life. What is quite interesting with that, it was a failure. It was actually no one was really interested in the tomato, not so much because it was a GMO, but because the flavor wasn't really that up to scratch. So that was kind of a premature kind of introduction into market of GMO products, but also obviously already starting to capture our imagination in regard to what are we going to be fed with. And then obviously a year later, the mouse with Oron spec came about. This is a product of tissue engineering. There was no genetic engineering in the process of making the mouse, but the public perceived that to be also a part of that larger kind of biotechnological project. And most people perceived that mouse with Oron spec as if scientists are sitting down and writing the code for a human ear and inserting it into a mouse, and the human ear pops up. And obviously that's not the case, but it did generate this notion that, yeah, we do have instructions of how to do things like human ears on mice from codes from the information side, which is not the case. But then a year after the mouse, we had Dolly. So Dolly wasn't really interesting visually, but it was an amazing technological fit, being the first mammal to be cloned. And again, the narratives and stories around that started to proliferate, and the public imaginary started to pick up. What's really interesting with Dolly is that because it was such an important technological artifact, museums didn't just collect the remains of the bodily remains of Dolly, but even Dolly's feces. So here we see Dolly's droppings that are part of the Wellcome collection, and they're being put on display once in a while at the Science Museum in London. But what, again, we need to mention, so first of all, you see the stage of the Petri dish, but you also see that we are willing to show a dropping of the cloned animal, but we're not, and there's no way you'll ever see any of the 277 failed attempts. They're not being kept, and they're not being presented as cultural artifacts, but sheet is. Then we, very shortly after Dolly, also we started to see the Monsanto Roundup Ready, the proliferation of large-scale GMOs. And again, those kind of stories started to proliferate around what can we do, what kind of power we have over other living systems. But it was still very problematic. So then it seems that only artists were moving into this area, while designers were stepping back until the area is more established. Or in a sense, maybe the artists were those who were presented those new things in a strange way as this idea of the freak show. And now we're starting to see the idea of the fact that we have a potential use of those technologies. So as I mentioned, I was very interested in tissue engineering. There's a really interesting kind of conceptual shift that happened between the 80s and 90s in regard to how we see bodies. So this image over here is from Life magazine from 1989. The idea there was to look at how we're going to repair bodies using hard, dry engineering kind of principles of electrical and mechanical engineering. Yeah, so every part of the body, the idea then was the body is a machine. Every part of the body is analogous to a mechanical solution that we can find, and therefore we can replace the body bit by bit by this hard, dry engineering product. That didn't really work as well. The body is a very corrosive environment. It's not very receptive to our ideas of engineering, and there was a realization that that might not be the path forward. And exactly 10 years later, so this is from Time magazine from 1999, so exactly 10 years later, you see the whole picture shifts to the body becomes its own technology. So first of all, the body is embedded with the information for its own repair within it, so we can somehow use it. We can somehow start to think about harnessing those latent regenerative powers of our bodies and start to create those body spare parts, either within or without or outside of the body, but using biological techniques. So in a sense, the body becomes its own technology. And so I kind of posed the question, what was quite interesting. So in 1995, as I was working on my thesis, there was an international consortium of scientists that claimed that in 10 years' time, they'll be able to build a fully functioning heart outside of the body, or grow, sorry, a fully functioning heart outside of the body. So I was saying, okay, if in 1989 we were told we can replace the body, or the heart, sorry, with a pump. In 1995 we were told that we can grow hearts. My question was, why not just grow pumps? And that was kind of where I started my own research, where I was looking at this idea. So this is kind of the very basic notion of tissue engineering here. This slide is actually from Joseph Vacanti's presentation in the third tissue engineering meeting in Orlando. And the idea then, so kind of mid-90s, the idea of tissue engineering was fairly basic, where you start with a biodegradable polymer scaffold that you create into a three-dimensional object in the shape of the organ you're trying to replace. You then seed this scaffold with the cells of the organ you're trying to replace. You grow it in vitro for a while, so outside of the body, and then you implant it into the body, and supposedly you have a new body part, a new spare part. This is how the ear on the back of the mouse, for example, has been done. But I wasn't interested in ever reintroducing those fragments of bodies, those functional tissues, into a body. I was really interested in the idea of what we then later on refer to as semi-living, those fragments of bodies that are being grown and maintained alive using technological support. So the idea of tissue engineering and regenerative medicine was around, as I said, growing new organs. So new organs, there were technologies that were kind of converging in regard to that. So stem cell research, cloning, and therapeutic cloning was pushed into this field. But at the very same time, there was or I started to recognize, and obviously it seems to be widely recognized now, that this very same technology can be used for non-medical applications, everything from research models to sensors, actuators, food and leather, and the use of tissue engineering for artistic purposes. So my original thesis from 1996 was called Living Surfaces, Biotechnology in the Design Way. It was a total speculative project, but then I decided to put my money where my mouth is and go and see a scientist and see if I can use or can start to work with those very same technologies that I was interested in. And to my surprise, I was invited into the lab. Yonat Zur joined me shortly later, to work with me on what we called the Tissue Culture and Art Project, where we started to look at the use of tissue engineering as a medium for artistic expression. Something which was striking for us from the very, very beginning was that our language in regard to life is very poor when we start to experience life in the lab. So you can see here, it's a half rabbit head that Yonat is basically taking the eye from. The first lab we worked in was an eye research institute. So you see something which is basically totally dead, and we then took the eyes off from that basically piece of dead meat. The eye itself was then put in an antibiotic solution overnight, and only 24 hours after the animal itself died, and I should say that the animal, as far as we know, all of those animals have been killed for food purposes, and the leftovers were sent to different research institutes around Perth, where they would take the parts of the body that they were interested in working with. But what you have here is basically the cells from the very skin that grows over the eye of the rabbit that we only started culturing more than 24 hours after the animal was dead. So we started to realize that our definitions and our understanding of life and death is extremely lacking when it comes to manipulating life within lab context. Obviously, scientists have been working on that for decades, but within a cultural context, there was very little discussion, very little ways of articulating the existence of those living entities that are living but not living in the same way as the animal from which they were taken. And obviously, in this case, the animal was well and truly dead, but the cells were still alive. And that's when we started to talk about in terms of the semi-living. The original projects we've done, or the very first project, was basically to roll the skin over the skin from the rabbit's eyes over different types of glass figurines that represented different modes of or different states of technology. We then developed other kind of what we refer to as symbolic gestures. The first living tissue engineered sculptures that we were able to present was in the year 2000, and there were semi-living worry dolls, which we presented living at the foyer of the main opera house in Linz in Austria as part of Ars Electronica. And what you can see here is we actually had to establish a fully functioning tissue engineering lab in this non-specialized environment, in this cultural place where we were growing our tiny worry dolls. Now, in retrospect, obviously, I can claim that the majority of the people that experienced this work didn't even see the worry dolls. This is a great example of where the technological frame takes over. So what we have here is basically the technology that is used to support the life of our artworks, if you like, is what people see most and way more than the actual worry dolls, which were tiny and very humble. Okay, but we were trying to different kind of methods of presenting the fact that they exist and that they're alive. So we would do things like feeding time in the zoo. Every day we would go and feed our sculptures and change the nutrient solution for their survival. I'll skip. We then kind of redesigned labs. So by now I think we built something like 30 or 40 labs in non-specialized environments. This is from the Science Gallery in Dublin. You can see the worry dolls being presented, this time kind of framed between a portal or a window over here, but the rest of the lab was a functioning lab that serviced the rest of the exhibitions or the rest of the projects in the show itself. We also recognize the fact that our sculptures are alive, but they're not going to be so by the end of the show, so we started to develop all of those killing rituals. So by the end of the show, or when there was no one there to look after our sculptures, we would kill our sculptures by inviting the audience to come into the sterile environment of the lab, take our sculptures outside of their sterile environment, and get the audience to kill our sculptures by touching them. I'm aware of the time, so I'll just briefly talk about the in vitro meat project, because we then moved into a series of what we refer to as pseudo-utilitarian projects. The first time that we grew a piece of meat was in the year 2000. Jonathan and myself were research fellows in Harvard Medical School at the tissue engineering lab there, and we basically used leftovers from another research that took place, which was on in utero skeletal muscle tissue engineering project with lamb cells, muscle cells. So we just grew those muscle cells into something that looked very much like a piece of meat. We couldn't eat that original piece of meat because it was a biomedical research lab, so there was no license to grow food. So in the 2003, we were able to do so by basically staging. We were part of a larger exhibition called L'Art Biotech in Nantes in France, and we basically staged this durational performance where we basically grew muscle cells from frogs for the 3 months of the exhibition, and at the very last day of the exhibition, we served it as the ultimate nouveau cuisine dinner. We grew about between 5 and 10 grams of the meat, and we then shared it among 6 people. And to our delight, 3 out of the 6 people who joined us for that dinner spat those pieces of meat out. The texture was, the sauce was really nice, but the texture was horrible. The polymer that we were growing the cells over didn't degrade completely. The muscle cells that we were using were frog muscle cells, but we didn't exercise them, so they were very much like jelly. So the texture was very much like trying to chew jelly on fabric. So 3 out of the 6 people that joined us spat the bits out, which was really great for us because we could then show them in the follow-up exhibition called The Remains of Disembodied Cuisine, which had a video documentation of the work as well as the bits that were spat out by unsatisfied customers, if you like. All right, skip that. We did do. I have been doing quite a lot of work around in vitro meat since the kind of basically a decade after we've done what we've done, growing the meat and eating it. This guy over here, Mark Post, unveiled his in vitro burger, and here you see him. I invited him for a cooking show, a cook-off, where he had to cook against a philosopher. That was before. That was 2012, before he unveiled his burger. So this is kind of a nice case where science imitates art. Yeah, so those are some images from that. Also, we found ourselves co-presenting. So this is from a show in Dublin in 2016, where again, it's kind of interesting that he grew two burgers, one that he served as part of his performance in 2013, and the other one he plastinized as a museum-ready piece that was then given to the Museum of Science and Technology in the Netherlands. And it's doing the tours now as a cultural object. No one can really chew on a plastinized piece of in vitro meat, but it's kind of interesting to see that from the very beginning it's already been positioned as a cultural object as much as it was for a scientific or technological object. So again, trying to capture the public imagination by the creation of those evocative objects. And that was presented alongside work that we did with an amazing designer called Robert Foster, where we designed a domestic bioreactor for the production of insect soup. So the idea was to actually maintain and use the nutrient solution as a way of being part of a soup where in vitro insect cells are growing as well. And the way it was presented was with this dark cloud of resources and nutrients that was hovering above this domestic instrument. Yeah, so we were trying actually to expose some of the resources that are needed in order to grow meat in the lab, as opposed to hiding it in a way that's just showing you kind of a nice, neat, plastinized burger, which was the approach that Mark Post took. We also did a victimless leather piece, where in 2004 we grew leather, and we then decided to present it actually not just as leather, but again showing all of the technical apparatus, the techno-scientific body in which the leather grows. When we presented it as part of the Design Elastic Mind show in MoMA in 2008, we had to turn it off, or Paola Antonelli had to turn it off halfway through the show, because we were using mouse embryonic stem cells that grew outside of control. And that really generated some interesting conversation. So the Design Elastic Mind was a fairly optimistic show, but the only living thing that existed there had to be switched off and be killed. And it shifted the conversation to something which I felt was way more important. That's the responsibility that we have towards those life forms that we are involved in manipulating. And obviously the media had a field day. New York Times had a headline, "MoMA kills art," and so forth. But I think the conversation was generated was quite important. We had a lot of really strange things happening to our victimless leather in the different stages we presented it. So it's always nice to see, and it's kind of nice to see what happens when you kind of not trying to control those things. Obviously, we set all of the situation to grow leather, but sometimes we grow some other things. I'll just skip all of those projects because I think that we are running out of time. But I just want to finish with that. So hopefully you know about the Grantor hype cycle, and this is kind of where the situation, or where I feel the situation is at the moment, where there was this technological trigger. And in the case of biodesign, it's really interesting. It's not so much a technology trigger. It's really about a conceptual shift in our understanding of what we can use living systems for and how we can use them. Yeah, so many of the technologies that biodesign is employing has already been employed either within kind of the biomedical research field or within agricultural fields. But the idea of kind of moving towards the development of consumer products is a fairly recent occurrence. And again, I would claim that first you had the science, then you had the technology, then you had artists coming in and kind of trying to make sense of it, and the designers follow, and the companies are following suit now. Yeah, so the technology trigger is really this conceptual shift, and we do see kind of as climbing now towards the peak of inflated expectations. So this is where the hype is in its kind of in its top, but then there's the painful kind of collapse down to the trough of disillusionment. Yeah, so you start to realize that many of those promises are not going to be fulfilled. Many of the promises are totally unrealistic and based on fantasy. And then this technology can either go down Death Valley, so basically nothing will happen with it, or it will slowly go up the slope of enlightenment into the plateau of productivity. So I've been doing this stuff for way too long. I've been thinking about it for the last 20 odd years. So I really see myself now very down the trough of disillusionment. Not really sure. Actually, some of the technologies I'm quite certain are going to face Death Valley, while others might slowly climb up the slope of enlightenment. And you know, you're here. You're just climbing up that towards this peak of inflated expectations, and it's good as long as you carry your own parachute. So I'll leave you there, and I'll be happy to answer questions.
Hi.
All right.
Okay. All right. I like how where we are. So we're about. I guess I got started in this about 10 years ago. So we're about 10 years, 10, 15 years behind you. Um, so a number of questions. I'll start with the last one. Oliver asks, what's next for you, Oren? What new things are you thinking about? What's exciting you? What should we expect from you in the next 5 years?
All right, there's two versions of this answer. First is like, you know, I keep on doing stuff. So as you mentioned, we have work at the moment at the Science Gallery in London, which is part of a series of incubators. So I became, United myself became very interested also in not just the living systems we're working with, but this technological apparatus that is being used to keep them alive and maintain them. So we developed in a sense, an off-grid incubator that is driven by compost, and it's kind of nice to have. And so we have it at the moment in basically just down outside the Science Gallery, just next to the Shard in London. And we have a big pile of compost, including some horse manure from the mounted police, heating up a bunch of mouse cells that are growing outdoors. And so as far as we know, this is kind of a really interesting case where you have an outdoor incubator where mouse cells are actually growing. But it's also kind of pointing attention to the resources and the apparatus that is the technological apparatus that is used to grow things outside of the original context. And that leads me to the point that many people that are moving towards biodesign are coming from an acute, and this was my original kind of education as well, from a very strong kind of eco-design perspective, the idea of being able to try and solve problems in the world by not just trying to imitate living biological processes, but actually using living biological processes, that there is an assumption that by doing so, you're more compatible with kind of environmental concerns. Yeah, so if you use a living system that is part of the ecosystem, you, in a sense, you're going to create good rather than kind of more drain on resources. But what we see time and time again that's not really the case. That actually some of the worst environmental disasters that we had are to do with concentrating biological systems outside of the context from which they were taken. Yeah, so if you think about factory farming and the huge environmental footprint that industrial farming has, and especially when it comes to cattle, for example, you start to see that not every use of living biological systems is necessarily an environmentally friendly one. So that's one, and we are going to do a series of works around it. I also, last year was the year of Frankenstein for me. It was the bicentenary of the publication of Frankenstein, and as part of that, I've been following Victor Frankenstein's footsteps and actually going to places where the book mentioned that he was, including the Orkney Islands in the north of Scotland. And I actually managed to find some material evidence to this fictional story. So we're now working on a documentary that's to follow that story of how they are. We basically were able to find and set up a puzzle of kind of putting things together to prove that some of the stories which I totally aware are totally fictional can be backed by material evidence, which is a very strange thing to think about.
Okay. Here's a follow-up question on the Gartner hype cycle. Well, I'm going to ask two questions, including yours, Iram. One is, how many times have you gone through this hype cycle? You know, you mentioned the first one in the or you mentioned one in the 90s. Certainly, there was one maybe 10 years ago. We keep seeing these booms and bursts.
Mm.
And then the next question following from that one, which is Iram's question, which is, what do you see going down the valley of death? What do you see reaching towards this plateau of
Right, both are great questions. So yeah, biotechnology is an interesting case because actually the first hype cycle was in 1970s, and I'm sure most of you are too young to remember it, but that was the first time that a living organism was patented, and there was also the birth of recombinant DNA and obviously genetic engineering. Some of you might be aware of the Asilomar meeting of scientists that actually was kind of a really interesting point in history where scientists were declaring a moratorium on some aspects of genetic engineering, highlighting some of the issues around it, but also using that as a way to allow self-regulation of those new technologies with an understanding of some of the issues that we're facing with unsafe use or with kind of unintentional release of GMOs into the environment. What was really interesting then is that that's also when insulin production was started to be developed using transgenic organisms. So there was a recognition of the fact that there is a market opening up and there's ability to produce different types of substances using those technologies. And so the 70s was a huge investment into biotechnologies that never really delivered, and that really chilled out the market significantly for about 2 decades. Yeah, so it took a really, really long time for the kind of biotechnologies from a kind of commercial and industrial perspective to recover from that initial shock of the 70s. Then we had obviously the Human Genome Project and kind of the late 80s, beginning of the 90s. But you should remember that all of those were much more engineering and biomedical. Designers and artists were not really involved in that. What's really interesting now is how, and this is going back to my original point about those social contracts, how artists and designers are actually being used in order to sell some of those fantasies. And obviously this Silicon Valley style startup culture didn't exist in the 70s as much obviously as it is, and this idea of trying to translate the very same mindset of Silicon Valley into biotechnologies and food systems is something that actually I wrote quite a lot about, and it can be problematic. So yeah, but I think we'll see some more of those cycles. What's quite interesting also is how all of those different technologies are now being clustered together. So when you go to the biofabricate meetings, for example, you would see people talking about everything from trying to make biocement using kind of different type of bacterial processes to people trying to grow leather and meat using mammalian cells and everything in between. So it's a very wide range, and this is, I think, the problem. And going back to the next question, that those things are clustered together. Some of them I can see that actually has a potential. So things like mycelium-based materials, I think, have quite a strong potential to actually be out there in the world and be viable. There's a lot to do with and a lot to say about the problem with upscaling biological systems. Mycelium seems to be doing quite well. One of the main issues that you always need to remember, if you want to grow things, you want to provide favorable conditions for what you're trying to grow. And in doing so, in most cases, you also invite other life forms to enjoy the bounty of resources that you provide the life forms that you're interested in. So mycelium is kind of interesting because mycelium can actually grow in places and thrive in places where other life forms don't really like it so much. So that gives you the ability to upscale that system. But saying that, actually Phil Ross presented in Biofabricate last year the fact that there was a patent application in regard to using mycelium to build bricks back in the late 19th century. Yeah, so obviously people were thinking about it, people were trying to do it before. It didn't work for them then. We're not sure if it's actually going to work now, but I would put my money, if I need to put, I think the mycelium area is actually an interesting area. Using animal cells and tissues for everything from in vitro meat to leather and any other kind of products, I wouldn't put my money on that as a large scale mass-produced consumer product. There's many issues that are there, and I don't think that they're going to be resolved anytime soon. So I wouldn't put my money in vitro meat, for example. Maybe those plant alternatives might be better. Then things like bacterial inks and all of those other kind of bacterial byproducts, and I suppose one of the things that I have the biggest beef with is all of the bacterial cellulose. I don't really think those are going anywhere. Are you there? Yeah, cellulose is the second most common biological material out there. We don't really need more of it in the world, so I don't really see any way in which the bacterial cellulose is going to, unless you somehow have an added value to it in some different way. But at this stage, all of those kombucha bacterial cellulose items are nice oddities and nice novelties, but I don't really think they're going anywhere.
I know we only have time for one more question, so let's make it a big one. You talked about 21st century, I'm sorry, 19th century institutions, 20th century ideas, and living in the 21st century. What do you think are the 21st century, what will be the 21st century ideas about life? And I'll extend that question. Do you see us having lots of different terms for living things? You use the term semi-living. Are we going to have a gradient of things that today we identify as just living?
I think it's extremely needed. So I always like to use this analog to the fact that in the English language we have 50 different words to describe shit. Yeah, so we're really good in giving kind of nuanced description of what comes outside of our body, but we're extremely bad in talking about something like life with all of its manifestations, and we have this really blunt instrument of this one word to describe all of those different manifestations and this kind of continuum. So yeah, I think one of the most important things and one of the most urgent things is to try and develop a much more nuanced language to describe what life is. Now, this is a huge ethical undertaking as well, because by giving those different names, you would also ascribe some value to them. And this is where the interesting experiments would take place, how you really engaged in developing a new language in something which is so charged, like the concept of life, and how you break it down. But I think that's going to be very needed, and I think designers and artists have a role there, because language is not just verbal, so we need to figure out new visual language to describe something that we don't have. I talk about in terms of the fact that we don't have a cultural language to describe what's happening at the moment. When you talk to scientists, obviously they seem to be very specific, but even then, when you talk to them about the fact that there's life forms that fall outside any form of a taxonomy, any form of classification, all of those cell lines that exist only as catalog numbers within tissue banks, those are problematic or kind of and very odd life forms that even scientists can't come to terms with as much. They're being used by them as tools, as research tools, as production tools, but within the larger scheme of kind of trying to figure out what place they have in the world, even many scientists would struggle to tell you where they are. So yeah, I think language is important, kind of creating new imagery to both capture our imagination but also question our imagination is extremely important. I would also like to argue that both artists and designers shouldn't fall down the trap of only becoming a kind of fantasy creators for this hype cycle. Yeah, we have to be way more sophisticated than that, and that's why kind of the ethos of what we have at Symbiotica is getting people to come and learn the techniques themselves. So what we do at Symbiotica, we run a lab where we get artists and designers learning the skills and the techniques in order to engage in the manipulation of life by themselves, rather than relying on secondhand evidence or kind of stories from scientists and technologists. Yeah, and that allows you also to understand the limitation of those technologies and some of the issues that are problematic. So we need to kind of also kind of blow open this black box of biotechnology and allow people like artists and designers to actually have a much more nuanced and rigorous understanding of what's involved. And I think this is really the important thing, and that's why I think Biodesign Challenge has a great potential there. But it's really about not becoming a fantasy factory, but really a place where people can engage and understand the technologies.
Oren, I want to be cognizant of your time. It's 8:03, and I know some kids have to get to school.
Yeah, I have a kid to take to school. Yeah, yeah, that's right. Yeah.
So thank you
Cool.
very.
But I'm happy to answer questions later on, maybe via my Twitter. So my Twitter handle is oron.cats. Yeah, you can find me.
Right. We'll reach out to you there. Thank you so much for doing this with us.
All right, cool.
And thank you to the audience for joining us
No
and
worries.
all your questions. I hear some projects in there, so if you're thinking about what you're going to present on stage at MoMA, in June, Oren presented a lot of interesting avenues to explore. So something to consider. Thank you, Oren, so much.
I'll have to say goodbye.
Thanks, everyone.
Thank you, everyone.
Bye.
Take care. See you soon.